Hello everyone, to all my dear doctors who are studying microbiology with me. Welcome to 𝗖𝗵𝗵𝗺, in which for the very first time in the history we are actually giving you lectures as per the system. And the topic of discussion today in microbiology is CVS and blood. We are done with the bacteria which causes CVS and blood infections. Now we are on the viruses. Okay. So this is class number 9th, and today the topic of discussion is HIV and AIDS, right? Myself, Dr. 𝗵𝗵𝗺𝗺, and I am your microbiology faculty and mentor. So my dear friends, coming on to HIV, please see here carefully that the structure of HIV is itself a very important question. And can you see this light green color? This is the outer envelope, right? So the HIV is an enveloped virus. Please, whenever you are studying anything, for example, now you are studying virology with me, you should go back and check the general virology videos for sure. I have covered it as per class number 10th and 11th and even 12th. So you can skip class 10th, but 11th and 12th are most important for general virology, because then only will be able to understand that what exactly is the meaning of the terms I am going to use in this class. Okay, so basically, if you can see here, this is the outer envelope, light green one, that says that HIV that belongs to the family Retroviridae, because it is a retrovirus. So generally what happens, DNA is converted into RNA. Generally what happens is this is called as transcription. But here they have got enzyme which is called as reverse transcriptase. So the transcription here is reverse also. That means the RNA will again convert into DNA. That's why retro, that means ulta, piche, backwards. Okay, so this is what is happening here. So this is the function of enzyme reverse transcriptase. That's why HIV belongs to Retroviridae family. Okay, so Retroviridae, I have already told you in your general virology classes that it belongs to the category where the organisms are what? Organisms are enveloped. Okay, so Retroviridae, it belongs to enveloped one. Okay, so see, this is the outer envelope, and after that you have got this yellow colored ones as transmembrane proteins. Okay, and then this red color one is the core which is there. Coming on to the materials, so if you remember, I told you the mnemonic for RNA viruses, and in RNA too I have told you the different examples, and in RNA viruses Retroviridae is one of the examples. Okay, so this is a RNA virus. Okay, Retroviridae is one of the examples of RNA viruses. Okay, and obviously some enzymes are going to be there. So this is roughly the structure of HIV. So family, as I have already discussed, Retroviridae, because they have got an enzyme called as reverse transcriptase. These are single stranded positive sense RNA. These are single stranded positive sense RNA. Okay, so if I just try to show you the different examples which I have taken in your introductory class, see here, Picornaviridae, Calciviridae, Coronaviridae. Then you have got 𝗲𝗲𝗵𝗺𝗺, Flaviviridae, HIV that belongs to Retroviridae. So all these are positive sense. The mnemonic also I have mentioned in the general virology class. So you can quickly go there and please do revise general virology before discussing any viruses, because repetition I am not going to do. It's your responsibility to go back and revise the topic. Okay, also these are enveloped viruses, also these are icosahedral symmetry. Okay, these are all things we have discussed in general virology. Coming on to mode of transmission, so remember that AIDS specifically has got a sexual transmission. Okay, so mostly heterosexuals, right, are the ones who are transmitting. Not heterosexual actually, mostly the homosexual people are having more chances of HIV and AIDS, right? And yes, but the most efficient way of transmission is blood transfusion. This is most efficient way. Mother to child transmission is also there, and sharing the contaminated needles is also one of the cause in transmission of HIV and AIDS. So please understand that AIDS is going to transmit how? For example, if I make a diagram for you, this is a contaminated needle. Needle is going to the blood vessel, right? So one is going to be your needle, other is blood vessel. Blood vessel means blood transfusion, okay? And supposedly this is the blood vessel of a mother, so mother to child transmission is also there. Plus one more thing which you have to understand is father mother. So you can remember like that you have got sexual transmission as well for this, okay? So this is the mode of transmission. Coming on to the types of HIV. So HIV has got two types, HIV-1 and HIV-2. HIV-1 is globally very common and it has got different clades like A to K. It's just the subtypes, okay, you can remember. But the problem with HIV-1 is that it has got a very high resistance. So any drug, if you are starting, the resistance is very high. And in HIV-2, it is mostly seen in the West Africa, and GP36 is the marker. Now how to remember? See here, HIV-2, this is 3, this is 6, so 3 into 2 is equal to 6, okay. So this is the HIV-2, so 36 GP, GP means glycoprotein. So glycoprotein 36 is the marker for HIV-2. Coming on to what are the clinical features? So generally it will have fever, lymphadenopathy and opportunistic infections. Now all the opportunistic infections are very important. I am going to teach you one of them in fact every opportunistic infection in some detail. So for now just remember fever, lymphadenopathy and opportunistic infection. So in laboratory diagnosis I have already discussed what all points are supposed to be remembered about HIV in general virology, okay, while discussing the laboratory diagnosis. Some very important ones I am discussing here. First of all, I have told you that viruses can't be cultured. Mostly viruses can't be cultured, but remember they can grow on the cell lines, okay. So some specific cells they require and they can grow in them. Why? Because this point also I mentioned in the introductory class that viruses are intracellular, obligatory intracellular, okay. So that's why they require some cell, or we call it as some cell lines to grow. So which cells are required by your HIV? Peripheral blood mononuclear cells, okay. So peripheral blood mononuclear cells are required and interleukin 2, okay. So peripheral blood mononuclear cells and interleukin 2, both of them are required for HIV to grow, okay. So remember HIV was HIV-1, HIV-2. So this is just a trick to remember. 1 means mono, so it requires mononuclear cells, right? And from 2 you can remember that interleukin 2, okay. So this is a trick and a mnemonic to remember. Coming on to antigen detection, okay. So you reached till interleukin 2, now you will double this, 2 into 2 is equal to 4. So 24 is the new number you are supposed to remember. That means P24 is the antigen which is required for the early detection, okay. So P24 is the antigen which is required for its early detection. Also one more thing you can remember that 𝗖𝗵 completed my MBBS in 2024, okay. So you can remember like that. P24 is the one who is going to detect. So I am a detector, not a dictator, detector, okay. Like that you can remember. Antibodies, how antibodies are detected? Using ELISA, using rapid tests, using the Western blot, which is the best confirmatory technique. So Western blot, in Western countries, I'll write it here, in Western countries people love to have pizzas, okay. So Western blotting is required for P for proteins, okay. So protein detection, because all the antibodies are proteins, so in order to detect the antibody, the best confirmatory method is going to be Western, okay. And then you have got something which is called as triple test strategy, which we use for AIDS, okay. Triple test strategy which we use for AIDS, okay. I'll discuss with you what is triple test strategy, but before that let us understand the other molecule tests. So for example, the baby is less than 18 months old. So in a less than 18 months old baby, we use DNA PCR, okay? But in order to check the viral load, we use the RNA PCR. Why do we check the viral load? To check for the prognosis. If the viral load is very high, we have to check how much RNA is present, but in order to diagnose it, we use DNA in less than 18 months. So remember, D for DNA, D for diagnosis. And prognosis has got P and R coming together. So R for we will check the RNA. Okay. So viral load, how viral load is calculated? Again, RNA copies per ml. Okay. Viral load, how to calculate? RNA copies per ml. Also, we have got one more thing that is called as CD4 count. That is also a prognostic marker which was being previously used. So in order to have the prognostic marker, another prognostic marker was CD4. Again, R is coming. Okay. So CD4. So this is important R. Again, R is important RNA. So RNA and CD4, both of them are for prognosis, and for diagnosis we use the DNA. Okay. So now let us understand about the genome and the different proteins. Okay. So please understand, you have got GAG gene, environment gene, ENV gene, and pole gene. So there are three genes which are there: GAG gene, ENV gene, and pole gene. Okay. Now GAG gene is responsible for P24. Okay. GAG gene is responsible for P24. Now how to remember? Everybody knows that initially we used to have which network? 4G, isn't it? So 24 is related to G. So G means GAG. Okay. That is further there in the capsid, and P17 is there in the matrix. Okay. So P24 is in the capsid, whereas P17 is there in the matrix. Okay. And in the matrix. And after that, can you see ENV? ENV is having GP160, GP120. So GP120 is there, leave GP160. GP120 is on the surface, and GP41 is the transmembrane, that is the transmembrane protein encoded by ENV gene. Okay, coming on to the next one, pole gene is required for the reverse transcriptase synthesis, integrase synthesis, protease synthesis. So imagine a person walking and bangs his or her head with the pole. So he is going to die, RIP. That means the pole gene is required for RIP, that is reverse transcriptase, I for integrase, P for protease. So pole gene for RIP. GAG gene, I have told you 4G network, so 24 and 17, whereas 120 and 41, these are big numbers. Okay, 120 and 41 are big numbers. So remember E for extra large numbers, extra large numbers. So GP 120 and 41, these are big numbers. Now 120 is very big, 120 is very strong, so it is on the surface, whereas 41 is big but not very strong. So 41 likes to be there transmembrane. Okay, it likes to be in the transmembrane. Similarly, if you see 24 is encoded by G, so 24 is there on the G and C. I always say go together because they look very same. So that is there in the capsid. Okay, whereas 17 is there in the matrix. 17 is there in the matrix. So this is about the genome and the proteins. Now coming on to the life cycle of HIV, very simple. So see here, this is the host and this is my HIV virus. So first of all, they are going to get attached with the help of what? With the help of a receptor. Which receptor? CD4 receptor. After that, there will be there will be the entry of RNA plus capsid. Okay, so the RNA and the capsid protein, both of them are going to enter inside. Then can you understand, the RNA is coming inside. Okay, once the RNA is coming inside, the next thing which is going to happen is that this RNA is now converted to DNA. The RNA will be converted to DNA with the help of an enzyme called as reverse transcriptase. So, reverse transcription will happen and there will be formation of DNA. Now, this DNA will be called as proviral DNA. Now, this proviral DNA is going to integrate with the DNA of the host. So, this is the host DNA. Okay. So, what is going to happen? The proviral DNA is going to get integrated with the host DNA. Okay. And then this host DNA, once it is integrated, what is going to happen? Now, there will be protein synthesis with the help of enzyme protease. There will be protein synthesis happening. So, protease will lead to the synthesis of protein. So, these are the different proteins which are being secreted or being formed. So, this leads to what? This leads to protein formation. So, this is the protein formation, and once the proteins are formed, they will be assembled. So, there will be assembly of protein which will be formed. Okay. So, now it leads to assembly of protein, and after the integration, viral protein synthesis, assembly, and this further leads to what? Now, the proteins are assembled. This leads to the formation of new virus, because the genetic material is there. Okay, the nuclear material, genetic material is there, the proteins are there, that leads to the formation of new virus. And because these are enveloped virus, this concept I have clearly told you in the general virology, that because these are enveloped viruses, they are going to show budding. Okay, all the enveloped virus, they show budding. So, new virus is going to get released by budding. Okay, and this leads to the formation of new progeny. I will discuss it again with you. So, what I told you, first of all, it is going to get attached with the help of CD4, then there will be a release of RNA, and then RNA will convert into DNA provirus, then DNA, proviral DNA and the host DNA are going to get integrated, and with the help of protease enzyme, okay, integrated with the help of enzyme integrase. Okay, so can you see all the three? RIP is being used, RIP. Integrated, then the protease will lead to protein formation, and after that there will be assembly of protein, that further will lead to new virus formation, and in the end it is going to get released with the help of budding. So, this is the life cycle of HIV. Now, coming on to this very important previous year question, they asked HIV infects which cells? First of all, they infect CD4 T lymphocytes and macrophages by binding to CD4 plus co-receptors also. So, one was the CD4, other is the co-receptors. What are the co-receptors? CCR5 and 𝗖𝗵𝗵𝗵, CCR5 and 𝗖𝗵𝗵𝗵𝗵. Okay. So, these three things, CD4 T lymphocytes plus macrophage, okay, and in fact the CCR5 and 𝗖𝗵𝗵𝗵𝗵, which are the co-receptors, this is the main target of HIV. So, it will kill the macrophage T lymphocyte. So, definitely the immune system is going to get decreased. So, it is called as acquired immunodeficiency syndrome. You got it? Yes. So, immune system is going to get weak. So, obviously the infections are going to come. There will be fever, lymphadenopathy and opportunistic infections. Why these co-receptors are important? Because the pharmac people say that if you give drugs against this co-receptor, the pharmac people say that you should give some drugs which inhibit. So, these drugs do what? They inhibit the integration. They inhibit the integration of HIV with these receptors. Okay, they inhibit the integration. Okay, you got it? Great. Now, I think yes, everything we have discussed. Just the remaining one is triple test strategy. What does this mean? So, actually the triple test strategy, if we discuss in detail, that's really important nowadays, because this is how we are diagnosing HIV right now. Okay, and please one more thing, very important previous year question I will tell you. So, for example, if a person is having tubercular infection and HIV all together, because they go hand in hand, we have to start the anti-tubercular therapy first, followed by antiretroviral therapy. That means you have to treat the TB first, then you will treat HIV. Otherwise, it leads to systemic inflammation, or you can say it leads to SIRS syndrome. Okay, this is just a piece of information for you to remember. In fact, we keep a gap of 2 weeks. This is more of a domain of pharmacology. So, pharmacology faculty will teach you, otherwise I will get killed. Okay, so I will not teach in detail. It was just an important piece of advice, so I shared it with you, right? So, what is triple test strategy? Yes, this is my domain and this is something I have to teach you. So, for example, first of all, we do some A1 test, any test for that matter, Western blot or ELISA or whatsoever, we do A1 test. Now, the patient can be A1 positive or A1 negative. For example, he is A1 negative, the patient is said to be negative only. That means there is no HIV which is there. But for example, the A1 is positive, then you have to go for second test, that is called as A2 test. So now there are 3 cases. One, the patient is A2 positive, the patient is A2 negative. Sorry, there are 2 cases. So A1 positive, A2 positive, A1 positive, A2 negative. Yes. So if he or she is A2 positive or even A2 negative, that makes no difference, or no difference. Just you have to remember that for formality's sake we have to do A2. Because in both the cases, either this is case number 1 where A2 and A1 both are positive, or A1 positive and A2 negative. In both the cases, we have to go for a third test. This is called as A3 test. So in this A3 test, what can happen now? Now the possibilities are A1 positive, A2 positive, A3 positive. That means all 3 positive, the patient is labeled as positive, HIV positive. But for example, if the patient is A1 positive, A2 positive, but A3 negative, it is considered to be as intermittent. Whereas, if the patient is A1 positive, okay, A1 positive, A2 negative and A3 positive, then also it is considered to be intermittent. Okay. And in the end, if the patient is A1 positive but A2 negative and A3 negative, you will consider it as negative. Okay, clear? So this is how you are going to interpret. Now, for example, like I told you that in both of the cases, these are intermittent. So what you will do for intermittent cases? These intermittent cases have to be evaluated that if they are high risk, you have to repeat test after 2 weeks. But if they are not high risk, you consider them negative. Okay. So this is about the triple test. Don't worry, I will discuss the triple test once again with you. So see here, the triple test strategy says you do a test A1 positive. If A1 is negative, then it is negative. You have to go for A2. If A2 is positive, A1 is positive. In fact, if A2 is even negative, you have to do A3. If all the 3 are positive, it is positive. If A2 and A3 are negative, it is negative. But if 2 are positive, 1 is negative, or even if 2 are positive, 1 is negative, then you have to check it is intermittent. If not high risk, consider it negative. If it is high risk, you have to repeat the test after 2 weeks. Okay, you have to repeat the test after 2 weeks. Okay. So this was major discussion for HIV. Now we will discuss about the associated infections. So remember, we have got CD4 count. I have told you earlier it used to be CD4 FOUR. R is the PR prognostic factor, right? So please understand that what are the Initially, it was used as a prognostic marker, and we have seen that at different levels of CD4, we have got different opportunistic infections which are occurring in HIV positive patients. Now, for example, if you have CD4 count less than 500, then you have got these co-infections like Candida, like Epstein-Barr virus, like HHV8, like HPV, and what they are going to cause. Candida is responsible for causing oral thrush, so you will have a whitish tongue. EBV is responsible for oral hairy leukoplakia. HHV8 is responsible for causing a cancer that is called as sarcoma. HPV, human papilloma virus, is responsible for causing squamous cell carcinoma over the cervix, right? So please remember that these are important if the CD4 count is less than 500. So how to remember? C, D, E, F, G, H. So what does this mean? So CD, that means CD4 count should be how much? Should be F, that means 500. CD4 count 500. So you will have H. What is H? H means HPV, HHV. Okay. What was E? E for EBV. What is C? C for Candida. Okay. All these things you have to remember. Okay, you got it? Great. So after that, coming on to next part, this is the CD4 count less than 200. So if it is less than 200, now even more organisms. So obviously if it is less than 200, these all infections are going to occur. So CD4 less than 200, all infections less than 500 are going to come for sure. But what are the next things? Other things can be histoplasma capsulatum. That's a fungal infection. We are going to read in detail about the histoplasma capsulatum. Next is pneumocystis jirovecii. This is very, 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 very important. Please don't forget it. Pneumocystis jirovecii, very important. Then you have got 𝗖𝗵 virus. Okay, which virus? 𝗖𝗵 virus, which is going to be there, and dementia. So if you see less than 200 has 2 words infection. So 2 words infection means, okay, this is a mnemonic. 2 words means, first of all, histoplasma capsulatum, 𝗖𝗵 virus, pneumocystis jirovecii. Okay, and dementia. Dementia is di means 2, 2 means 200. So this is the way to remember these things. And very, very, very important thing which I have to tell you is that all the infections less than 500 CD4 count are going to come if the CD4 count is less than 200 for sure. But if it is less than 100, my dear friends, then Aspergillus, we'll discuss in detail about Aspergillus. First, Aspergillus is also going to come here. Bartonella is also going to come here. Again, very important. Candida is going to be there. Cryptococcus is going to be there. Okay. Then Cryptosporidium can be there. CMV can be there. Okay. So what will happen in CMV? It is going to attack the retina. Okay. And for your MCQ purpose, please understand CMV shows intranuclear owl eye inclusion bodies. Okay. So inside the nucleus, it is going to show owl eye inclusion bodies, but intranuclear. Okay. And you have got something called as Mycobacterium avium intracellulare. For now, you are just supposed to remember the names and the mnemonic. If it is less than 100, the mnemonic is A, B, C only. Aspergillus, Bartonella, then Candida, Cryptococcus, Cryptosporidium, CMV, and from this M you can remember MAC also, Mycobacterium avium intracellulare complex. The C is nothing but the complex. Okay. So this is the trick to remember less than 100. Coming on to the next part. Okay. So there are 2 more actually, EBV and Toxoplasma gondii. Toxoplasma gondii I've already discussed a little bit in your fetal infection classes in general microbiology, and other is the EBV. Okay. So you can do what? You can do like this: A, B, C, D, E. What is this? A I have told you, B I have told you, C I have told you, CD means CD4 count, and then E for Epstein-Barr virus. Okay. So this is the trick to remember all of them. Coming on to a little bit part from pharmacology, but then although the pharmacology sir or faculty will or ma'am will definitely kill me, but then still it is my responsibility to tell you the normal and abnormal things. Please understand now, the virus coming on and getting attached to the receptor, I have already told you that they are going to infect the T cells actually, and in fact the CCR5 as well. Okay. Then after that, once it is attached, what it is going to do? It is going to do uncoating. What is uncoating? Uncoating means the release of RNA. Okay, release of RNA. And this process, okay, so this process is actually something which requires some ion channels. Okay, so the process of uncoating, the release of this one requires ion channels. Okay, ion channels to work. After that, once the genome is released, it leads to the synthesis of protein, and once the protein is synthesized, these are going to have the assembly, and then it is going to get released with the help of budding. Okay, budding and release. So, what are the different drugs which are used? So, in order to inhibit the fusion, you have got a drug called as 𝗵𝗵𝗴𝗺. So, FU means fusion. Okay, FU means fusion inhibitor. So, fusion inhibitor means 𝗵𝗵𝗴𝗺. Then ion channel blockers. So, ion, you have I and N. So, I and N, I and N. So, amantadine. Okay, you got it. After that, you have got polymerase inhibitors. Okay, so you have got what? Polymerase inhibitors. So, acyclovir, efavirenz, 𝗲𝗲𝗵𝗿𝗺, these are polymerase enzyme inhibitors. Okay, efavirenz, 𝗲𝗲𝗵𝗿𝗺 and acyclovir. After that, you have got protease inhibitors which are having 𝗲𝗏𝗃 in the end. 𝗲𝗏𝗃. Okay, so protein. I think every Indian parent says, don't take protein. They say, na, don't take extra source of protein, have a healthy diet and your protein will be taken care of. So, don't take protein, or they say, never take protein. So, the mnemonic is never. 𝗲𝗏𝗃 is the protease inhibitor. Is what? Is the protease inhibitor. Then you have got neuraminidase inhibitor, and neuraminidase has got Mi word in this. Here also Mi is there, 𝗲𝗲𝗵𝗴𝗃 and 𝗪𝑒𝑙𝑡𝑎𝑚ivir. Okay, so these are the mnemonics for discussing the drugs, antiretroviral drugs. Now, important part, that is prophylaxis. Okay, so for example, accidentally or by chance, if some patient is coming in contact with HIV positive patient, then what is going to happen? Then the first line regimen for HIV post-exposure prophylaxis is tenofovir, 𝗵𝗵𝗏𝗏 combined with dolutegravir or raltegravir. This is followed in some places, but in most of the places nowadays they are following this tenofovir, lamivudine and dolutegravir. So, TLD is the drug regimen which is being followed. Okay, and to reduce the maternal fetal transmission risk, the WHO recommends triple antiviral therapy. So, triple antiviral therapy, I have told you, this is to be given during the pregnancy, and administration of intrapartum intravenous 𝗲𝗲𝗵𝗿𝗺. Intrapartum means during delivery you are supposed to give 𝗲𝗲𝗵𝗿 drug. It decreases the risk of maternal to fetal transmission. So, this is all about our discussion for HIV. I hope that HIV is, it is actually a little tricky topic, but with Micro Mahasutra and with our classes, you are able to retain this topic better. Thank you so much. My feedback section in the comment section is always open for lovely comments, and obviously your love, your blessings, your support and care. Thank you so much for watching. This is the end of HIV and AIDS topic.